Hello, everyone. Welcome to Northwest Michigan Orchard and Vineyard Show. Today, we are going to talk about many good stuff. First of all, I would like to say thank you to our sponsor. They financially help us to prepare this program and prepare those goodie bags for everyone. We are going to send some of them to Canada or other countries. So it's really good. So I would like to remind that Michigan State University providing equal opportunity for everyone, regardless of their differences and their gender or other differences. I'm sure everybody know how to use Zoom, so just I'm going to remind that how you can send your question or note. If you have any question or note, simply you can type your question in Q&A box and chat box and send it for us. And in audio setting, make sure you are choosing right speaker. And at the my special thanks goes to our collaborator. They also they help us a lot to prepare this program. And at the end of this meeting, I'm going to show RUP credits info. Make sure you write down those information. Thank you. Our next speaker, we are waiting for him. He's going to join us. He's Dr. Jose Ramon Urbez-Torres. He's from Agriculture and Agri-Food Canada. He's going to talk about trunk disease management.
Good morning, everybody.
Hello.
How are you?
Good morning.
Let's share my screen.
Thank you for joining us.
Oh, sorry.
I would like to give a brief introduction for you, if we could this morning. Welcome, everyone. It's 11:15. Thank you for joining the Orchard and Vineyard Show virtually this year. Our next talk is grapevine trunk diseases. What are they and why should we care? And how can we manage them? Our speaker today is Dr. Jose Ramon Urbez-Torres. He is a research scientist at the Agriculture and Agri-Food Canada Summerland Research and Development Center in British Columbia, and he serves as an adjunct professor in the biology department at the University of British Columbia campus. He received a postgraduate master's degree in viticulture, enology, and winemaking in 2001 from the International Social Science Council and the degree of agricultural engineering in 2004 from the University of Valladolid in Spain. He completed a PhD in plant pathology in 2009 at the University of California, Davis. Dr. Urbez-Torres has studied diseases of woody perennial crops since 1999, and his current research at AAFC focuses on the development and implementation of sustainable management strategies for fungal, bacterial, and viral diseases of grapevines and tree fruits in Canada. Dr. Urbez-Torres is the current chair of the International Council on Grapevine Trunk Diseases. Welcome this morning.
Thank you very much. Good morning. Can you see my presentation?
Yes, but it's not in presentation mode.
Yeah, can you see now? It's all good?
Yes, perfect. Yeah, thank you.
Okay, perfect. So, thank you very much. Good morning, everybody. I really thanks the invitation and having the opportunity to present this topic and part of my research. And today, as we have mentioned, I'm going to talk about what are trunk diseases, why are they important, and how can we control? That's the most important part. So to start my presentation, as I mentioned before, We will start with what are plant diseases, the different diseases and associated fungi that and the symptoms they cause. Why should we care? The significance and economic impact of these diseases that sometimes or many times, I have to say, they are overlooked in our vineyards until it's too late. And how can we manage grapevine plant diseases? And for this, I will make a strong point on the importance on understanding what we call the epidemiology of these diseases, pathogen inoculum sources and spread, and we'll touch in the most common chemical, biological, and cultural practices. So to start, what are grapevine plant diseases? Well, these diseases are caused by many different fungi. The majority of fungi causing plant diseases are in the Ascomycota group, but we have some basidiomycetes, as I will explain later, also being part of this complex. Grapevine plant diseases, these fungi infect grapevines primarily through wounds and openings. And as you can imagine, what we do every year in grapes is pruning them. So pruning wounds are the main point of infection. We are creating year after year point of entrance for these pathogens to come in the vines. But other types of wounds, either caused mechanically by mechanical damage or retraining in the vineyard can also be a point of entrance. And also we know that some grapevine plant disease fungi are thought to be latent pathogens, and I will touch a little bit later what this really means and the work we are doing right now to understand this factor. The overall symptoms include a slow or rapid dieback, and that will depend on the plant disease you have or the complex of fungi in your vines, and sometimes sudden vine collapse. There is a progressive death of the plant parts, such as spurs, cordons, and of course, eventual death of the entire vine. Grapevine plant disease occur wherever grapes are grown, and the disease and pathogens incidence may vary by geographical region. So environmental conditions and geographical region has importance on the type of plant disease and fungi you may have in your region. So grapevine plant diseases, we call disease complex or complex diseases. We can divide it in two main groups. In young vineyards, and young vineyards again will depend where you are located. But roughly within 5 years after planting, we have two main diseases: blackfoot disease, which is caused by fungi in this genera, Campylocarbon, Ilionectria, and Dactylonectria. And the main difference of these fungi within this disease versus the others is these are soil-borne fungi. These fungi are soil inhabitants. Then we have Petri disease, caused mainly by Phaemonia Candidospora, a species of Acremonium, with Acremonium minimum as the most important, and Cadophora luteolibacea. Until recently, we really believed that these were two separate diseases, but field work and isolations from plant material, you can find pathogens of both diseases in the same plant. It's not always easy to differentiate symptoms, so this is what we call today the young vine decline. And then in mature vineyards, we have ESCA, which includes all the fungi causing Petri disease. And here is where we can find some of the basidiomycetes I was talking before, this mushroom-like, that will be playing a role on ESCA disease. And then we have what we call the canker diseases, Botrytis dieback, Typa dieback, and Phomopsis dieback, causing these characteristic cankers and some other symptoms we will talk now. So in terms of young vine decline in young vineyards, some of the external symptoms, as you can observe here, as overall poor growth, poor vigor, short internodes, some of the sudden collapse or wilting that we were talking before. But as you can see, it's very difficult just based on these symptoms to associate it to a grapevine plant disease or to a young vine decline. These symptoms can be also caused by spring frost, or in our case, in some regions, by winter damage. So it's not always easy to identify just based on symptoms in the field. However, if we collect those plants, we can see very characteristic internal vascular symptoms in the plants. You can see here all this streaking, all the necrotic discoloration of the vascular system, completely rot of the base of the rootstock. Those symptoms are very, very characteristic of young vine decline, and the pathogens are usually isolated for all these necrotic areas. There is a very large number of different pathogens that can cause young vine decline. Here in red, you can see the most commonly found and the most important, but as you can see, there is a very, very large group of pathogens in this group. SCA is another important disease, and actually is the disease has been longest study, almost over 100 years of a study of these diseases. This disease was the disease that was first discovered actually in France. So we have what we call the SCA foliar symptoms, commonly known as leaf tiger stripes. So they form this intervenial necrosis that edge of the leaf will start scorching, and in the fruit can cause what we know the black measles. These black measles, these dark points here in a table grape, so some seedless, as well as here in a wine grape. This is a Cabernet Sauvignon. We can see those symptoms caused on the fruit. These are caused by toxins produced by the fungi that they get translocated, and for example, it's a big problem for table grapes as they cannot be marketed. Also, what we have on SCA is what we call the apoplexy or the sudden collapse. So vines, sometimes infected by SCA, in the middle of the summer, they will completely collapse, wilting off all the leaves, of course, with loss of yield. And it's interesting to mention that not always these vines will die, and some of them, they may recover the year after we experience a collapse. As I mentioned before, we have the pathogens that cause SCA, Petri disease, as we have been talking before, and here we include the basidiomyces, with Vomitiporia mediterranea as one of the main basidiomyces known associated with this disease. Sometimes we can see actually the basidiocards, the fruiting bodies of these basidiomyces on the vines, and they cause this characteristic soft yellowish bud in the vine. In terms of the canker diseases, in mature vineyards, we have Eutypa dieback, and this is one of the most characteristic diseases in the sense that cause very typical symptoms, which they are these short internodes, capped leaves with necrotic edges, and as we can see here, where we have the canker on the infection, we see the foliar symptoms, while in other areas of the same vine, the vine can grow normal. They cause these perfect wedge-shaped cankers. And in infected areas, if we look with a lens, we can see these small little fruiting bodies which are embedded. These are these flash-shaped fruiting bodies with the spores inside. Here we can see the spores of Eutypa lata, which is among, you can see all the different species causing Eutypa dieback in the genera of Cryptosphaeria, Cryptobalsa, Diatride, Archipela, Eutypa. Eutypa lata is the most important as well as the most virulent, and to date is the only species that has been shown to be capable of causing the foliar symptoms. The next disease is Botryopsphera dieback, and of course caused by, again, another large battery of different Botryopspheriaceae species. The main difference between Botryopsphera and Eutypa dieback is Botryopsphera dieback not always cause foliar symptoms. We can see here completely dieback of a cordon. We can see here that spurs and buds will not grow in the spring season. And if we make a cut here, we can see how the canker of Botryopsphera is inside, also causing some wedge-shaped cankers. And very interesting, we can see here in this part, the bottom left picture, a canker progressing in the cordon, but total normal symptoms in the plant. So this is something that's very characteristic with Botryopsphera, and for this reason, it's difficult sometimes to identify because we can have vines not showing any symptoms with the in our dead cordon. We can see also a streaking of the bud, and in some cases, so not only cankers, but also dark streaking of the bud. And in comparison, or in difference with Eutypa, we have these fruiting bodies, which are called pycnidia, and they are also embedded in the bark with a hole on the top, where the spores are released. And this is just an example of some of the spores of one of the species. And finally, we have Phomopsis recently identified grapevine trunk diseases, very similar symptoms to Botrytis dieback, with lack of spring growth in infected spurs. Some of these spurs, you can see these bleach symptoms, well-shaped cankers, and as well as pycnidia, same as Botrytis. So this is caused by the Aportean pelina, which was previously known of Phomopsis viticola, and as you know, because of Phomopsis cane and leaf spot, which is the foliar disease. It's a colony with the pycnidia and the cirri coming with the spores and the alpha and beta spores typical of Phomopsis viticola. There are other diaporte species associated to this disease, but this disease is really most prevalent in regions with high Phomopsis cane and leaf spot disease pressure. So, as I mentioned before, it's not always one disease in one plant or one pathogen in one plant. In many cases, we can say a large pathogen diversity, and in this case, in young plants. This is some of the symptoms, in young plants that we can see at the bottom of the rootstock. All these necrosis, very, very limited light vascular symptom. This is in the area, upper in the rootstock, and the streaking we have been talking before. So in this case, we conducted research here in my laboratory, and we developed a molecular macroarray that basically was able to detect in one simple test over 70 different pathogens associated with young vine decline. So as proof of concept, we collected plants from different nurseries, grafted plants. We collected 4 different types of samples: scion, graft union, at the end of the rootstock and the roots, and self-rooted plants, of course, all the samples without the graft union. These samples were processed and analyzed. And what we observed is ready-to-plant material evaluated from 2 nurseries give us all these different types of pathogens in the material. 92% of the plants have some black foot pathogen. In orange, you can see all the black foot pathogens. 70% of the plants have Botrytis. In red here, we have 60% of the plants Pneumoniella, Cadophora, and so on. So we can see the large diversity of pathogens that we could find in plants. And of course, many plants hosting more than one of these pathogens. Same is we look into mature vines. We can see that in many cases, symptoms overlap. We can see here wedge-shaped cankers with the Botrytis, that Ripaceae. Let's move into the second part, was why we should care about trunk diseases, the significance and economic impact. Basically, while I was talking about these diseases are not new, they are as old as vine cultivation. And as we can see here, historically, since the end of the 1800s, where ESCA was first described in France, later on, studies done in New York about dead arm disease. The symptoms of petri disease were reproduced in Italy in the early 1912. Description of tiger strip leaf symptoms in 1926. Botrytis was first known as causing cankers in Ontario in 1964, and Utah in California in 1978. So why the emergence of this disease? Why we are seeing this disease suddenly or in this explosion? So we have what we call the emerging diseases, that either have occurred previously, but affecting only a small population in isolated places, or have occurred through human history, but have only recently been recognized. So in the grapevine trunk disease community, we believe that this is one of the major factors that have contributed to see these days this explosion of grapevine trunk diseases. There are several hypotheses, mainly, planting booms that resulted in poor quality material, not necessarily to a huge increase on acreage worldwide, but vineyard replacement, change of varieties, or in some cases, mostly what we call the new world viticulture, where there was significant expansion of vineyards. Prohibition or phase out of effective chemical controls like sodium arsenate, was banned in 2000 in Europe, was highly efficient to control ESCA, or the phase out of venomil in 2001, which was basically one of the few systemic fungicides that we could control the disease once the plants were infected. We have also changes in viticulture practices, and probably these ones have played a major role, the mechanization. And just to give you an example, I wanted to show you here a vineyard from my hometown in Spain. These are 80, 90-year-old Garnacha vines, a very low planting density, very few spurs, with very few pruning wounds, still growing, low production, of course, but still alive and still producing. We are moving now mostly to this production system, double cordon, spur prune. Basically, in one vine, you can have, between 15, 20 spurs. If we multiply that for the number of pruning wounds, we can have up to 100 pruning wounds per vine. So we have significantly increased the chance of this fungi as well infecting plants. So there are important economic impacts of these diseases. For example, in California, cancers cost growers about $260 million a year. You type a dieback only in Syrah in Australia can cost over $20 million. We have also the issues with young vine decline that, of course, can cause important economic losses, mostly due to the replacement cost before even the vineyard reaches full production, and you can have a cash flow, and also the difficulty to manage an even vineyard. That we are going to have vines, growing 3-4 years ahead of replants, and that can cause significant nightmares for the grower. So worldwide economic impact was estimated in $1.5 billion US, and this is based on the replace only of 1% of planted grapevines. So in France, for example, it's the most important biotic factor limiting yield and vineyard lifespan, and basically about 12% of the current acreage in France is not economically suitable due to ESCA, which can result in losses of about 1 billion euros. So great work done by the economist Jonathan Kaplan in California. So as here the incidence by age of the vineyard of grapevine trunk diseases, and we can see here, of course, at the vineyard age, we are going to have a much higher incidence of trunk diseases. By year 10, you may have a 20%. By year 15, about 70%. This also reflects on loss of yield, and we can see that an infected vineyard by year 10, we can have a 20% loss of yield. Between 10-15 years, we can reach already a 50%, which obviously is unattainable economically for the grower. So, one of the questions, we have powdery mildew control since year 1. The first season, we start managing powdery mildew. Usually, sometimes, grapevine control, maybe year 6, year 7, or year 8, when we start seeing these symptoms in the vineyard. This is already too late, as I mentioned before. The fungi have made their way already into our plants, and there is no way that we can eliminate the infection inside the plant. And this not always occurs, actually. Sometimes it can be even much later. Just to give you an idea of the incidence, for example, in British Columbia, which is a very young and emerging grape growing region, and this is data from 117 vineyards, both young and mature. We can see here, every single vineyard incidence of trunk diseases, and here is the average by age. We can have here, a significant incidence in some of these young vineyards by young vine decline, reaching up to 40%. We have kind of a plateau, between 5 or 15 years. Mostly these vineyards are getting infected, and after 10-15 years, they start showing, a high disease incidence. Still, some of these old vineyards with no symptoms, but no symptoms, no foliar symptoms, doesn't mean that the fungus is not inside the plant, as we have seen before with Urbez-Torres dieback. So how can we manage trunk diseases? And I wanted to start talking about the importance of understanding pathogen of the disease, which is going to give us, the best information to implement proper control strategies. So the first source of pathogen inoculum is the propagated material. And this is not something I'm saying, but it has been well studied for a long time. This is a paper by the Spanish group, Gramaje and Arbegot, where it's a very nice, interesting review paper talking about, the incidence of fungal tram pathogens in the grapevine propagation process. And here there are some pictures, from propagated material. I saw before symptomatic, where we can find many of these pathogens inside. The grapevine parasites can infect, plants through the different steps of the propagation process, mother fields, the nursery process where there are hydration tanks, cold storage, disbudding, grafting, callousing. So you see basically any step we are having a wound or producing wounding in our plants, we have a chance of entrance by pathogens. And of course, nursery fields. And here is just a list of the different control strategies that many nurseries, try to implement to minimize the effect of the infection of these pathogens, whether are by the application of biocontrol agents or water treatments, fungicide dips, etc. But one of the things we have learned, as I showed you before when we were working on our identification of pathogens from young plants, is that some of these fungi can be identified from asymptomatic material. That's why we are calling some of these pathogens to be latent pathogens. So they may lie dormant here in the bud, not causing any symptom or any damage into the plant until they become virulent. And why they become virulent, I will talk later about it, but it's basically possibly a stress condition that make this fungi go from a latent phase to a virulent phase. So here we can see a picture where asymptomatic plants, we are able still to isolate different fungi. And from symptomatic, you can see here all these necrosis that we have been mentioned before. We are able sometimes even to isolate the same pathogens. So this just to give you an idea what will happen in the lifespan of a vine. So if we have here the death of the vine, and this is the time, a healthy vine will, of course, naturally decay and die with the pass of the years, but we have a much longer lifespan. If some of these vines, is infected with one pathogen and has some stress, is going to have a premature death. And our hypothesis also is if it's infected by more than one pathogen under stress conditions, the vine is going to suffer a premature death much earlier. So the key point is to really understand which stresses are making, these vines reaching this premature death when they are infected with some of the tram diseases. One of the things we have been working on is the implementation of a recently developed molecular tool, droplet digital PCR, to detect, and quantify pathogens within the planting material. The main difference of ddPCR versus normal PCR is we are going to have many thousand of discrete measures compared to one. Each sample is divided in many small droplets. Each droplets have a PCR reaction, so we are going to have a much higher chance of detection. So we have developed primers and probes for all these different pathogens, and we are testing the different parts of the plant. So first, here you can see for Salmonella chlamydospora, you can see, a significant difference on the inoculum concentration. Each of these dots, blue dot you can see here, is a positive reaction for one of these genes we are targeting. So you can see here in different plants, from no Salmonella at all to a high concentration of Salmonella. So a very large diversity of the pathogen depending on the plant. This is our positive control and negative control. So for example, a percentage of infected plants in this study, we have, for example, Chardonnay, 100% infection with Salmonella chlamydospora and Cadophila olivaceae. Very high also with Elonectria. Merlot, 100% with Cadophila olivaceae. So you can see that most of the plants were infected, and some of them were fully infected of the presence of these pathogens in the plants. So our hypothesis is that the presence of the pathogens may not result on disease development and eventual death, and is going to be dependent on the amount of inoculum present in that plant. It's going to be a threshold that the plant still can fight, and we are not going to see disease development. If presence of the pathogens means mortality of the plant, all these Chardonnay vines with 100% Pharmorilla and Cadophora, they will never have survived. And that's when we don't see that when establishing vineyards. We see some mortality, but not 100% failure. So there is also a significant inoculum concentration difference between cultivars and among plants within the same cultivar. So these are all 45 plants. So these are 15 plants of all 3 different varieties: Chardonnay in blue, Merlot in green, Pinot Noir in red. And we can see here the amount of pathogen from every single plant. In this case, we can see that Chardonnay plants have the highest amount of Salmonella chlamydospora. One plant of Merlot have a quite significant concentration, but then you can see that the majority of the plants have very low amounts of pathogen in them. So there is a very significant difference between plants. Also, we can see a significant difference between regions of the plant. In this case, Salmonella chlamydospora, inoculum concentration was much higher in the graft union. Then on the basal end of the roster, with barely detected in the roots, and we know that this is not the pathogen of the roots, and in the scion. So we can see also different regions where we can see different concentrations of the pathogen. So right now, we are working in our laboratory to try to understand and determine the factors that favor this transition from latent to virulent phase. This can be abiotic stress factors, water stress, nutrient deficiency, overcropping, poor planting, biotic stress factors, other pests and diseases. We are working right now with nematodes, inoculum tresors, and we are also working with AMF of mycorrhizal fungi and how they can help us to minimize this stress and therefore, avoiding disease development. So this is my student Jared, PhD student right now conducting greenhouse, both and field studies to try to give some answers to these questions. So this is the characteristic life cycle of grapevine trunk diseases, as I mentioned before. So we have the pycnidia, in the dead parts of the cordon. Pycnidia putting the spores that will infect ring wounds and so on. We can also get spores from alternate hosts. Grapevine trunk diseases are known to have a very wide range of different hosts, many different tree fruits and other plants, as well as native plant communities. We can localize those fruiting bodies, those pycnidia, in all parts of the plant. And this is electron microscope of some of these pycnidia embedded in the bark with the neck and the ostiole, the hole at the end where the spores are released. So this is just showing you, in this case, the perithecia of a Cremonium minimum that is actually releasing the spores. These spores are within a sac called ascus, and usually we have 8 spores per ascus. It's very common on ascomyces. So these sacs with the spores reach the outside because of a different pressure, the ascus will break and these spores will get released and they will go within the vineyard or also, some distances to other vineyards by wind, rain droplets, even insects have been shown able to carry these spores. So there have been significant amount of studies trying to understand, epidemiology around the world using different devices, spore trace devices, basically focus on identifying inoculum availability in the field, as well as low and high periods of infection throughout the year. This is going to be critical information, basically, to really implement and target our control strategies. So, for example, some studies conducted in New York and as well as in Michigan in 1980 by Tracy on Utaipa plata is showing the different periods of the year where spores were commonly captured in the field. Mostly in both New York and Michigan. The months between March, April, and May, here the same, March, April, and May, have the highest concentration of Utaipa lata spores. very well correlated, for example, here in Michigan with precipitation. Here in New York, we see that precipitation basically occurred throughout the year, and most of the spore release was between March and April. So if we have already this information in Michigan that, of course, at the end of the winter, early spring, though this is the critical time to be aware of protecting our pruning goods. Similar studies conducted in California, in this case, on Botrytis spore trapping, a little bit of a different situation in California, again, associated with environmental conditions. The bars here represent the total amount of spores. Blue is precipitation and temperature. So we can see that 60% or more of the spores were released from basically late fall to midwinter. Less than 20% of the spores released in fall or early spring, and only 3% of the spores released in late spring and summer. So basically, as soon as we are leaving our rainy months in California, usually January, February, spore release stops, and we have a dry period in California with no spores. This is one of the reasons why in California, late pruning is quite efficient, because if we are able to delay pruning until we have passed, the main source of inoculum, we are significantly reduce the infection in our vineyards. Similarly, we need to protect pruning wounds throughout, the dormant season, and as well, once we are out of this highly inoculum time, those wounds can be also protected, to the end. In British Columbia, we have done also studies that we have seen, again, a different pattern, and this is very important to understand that different regions we have different epidemiology of these diseases, and that's why it's significantly important to conduct these studies at the regional base. So here we have the total amount of spores through the study of 3 years. Here we have temperature and precipitation. So basically, in our conditions in British Columbia, we see that the first release of the inoculum happened, similarly, like for example, like in Michigan or New York, late winter, early spring. In our months of April, May, that's where we're gonna have inoculum started. We have a high release of spores about 6-7 weeks after our first inoculum, but you can see here that inoculum is present throughout the entire growing season. So we first are gonna have to be able to protect pruning wounds through a long time of this growing season, and we can see that inoculum start decreasing, and basically during the winter, there is a much lower presence of inoculum. Spores, in temperatures below freezing, in this case, 2 Celsius, they are not able to germinate. So that's why probably one of the possibilities that there is no pressure during this time. So I'm sorry, I'm supposed to be finishing right now, but because of the technical issue, I'm gonna try to go through the control strategies, and hopefully we have some time at the end for questions. So we're gonna talk a little bit about chemical, biological, and cultural practices. So as I mentioned before, there are currently no products, chemical or biological, capable to cure an infected vine. When the vine is infected with a trunk disease, we don't have a way other than what I mentioned here, remedial surgery to eliminate the disease. Many of you may be familiar with remedial surgery, and it's basically what we call the chain. So a cure where we can start following the disease and start cutting parts of the plant until basically we reach the end of the growth of the canker infection, and is usually advised to cut about 15 centimeters below the end of that infection, because the pathogen can still be there growing with no infection. And then we will retrain these vines. This is currently the only way we have to eliminate the disease from the plant. They have several advantages. We can retain the same clone and fruit quality. Sometimes, all vineyards where it's difficult to find those clones again. We have a very well-developed root system, so we will be able to come faster in full production. I see this type of work in California, and vines coming under production after a year. And of course, higher success if vines are self-rooted. Once the infection has passed through the graft union, the vines are basically condemned. Disadvantages is labor intensity and high skill. Workers are needed. It's costly. A study in 2008 estimated about over $2,000 per acre. And better results if done during the dry season period when we know there are not going to be spores in the environment. We are making very big cuts. And of course, it's very, very hard to do this work, during the growing season. It's mostly done during the pruning time when actually, we have the conditions that spores are released and can reinfect. So current control of plant diseases are primarily based on prophylactic practices. And of course, right now is pruning wound protection. So we have products registered, against Scaliotypa, Botrytis dieback. There are paste and paints, they are very effective, and also even more effective if a fungicide is incorporated. They cover very well the pruning wound, and it can give us protection for a significant period of time. However, they are hand applied and can be very highly costed to apply these products. Therefore, right now, the main request from industry is the development of products that can be sprayed. There is a possibility to treat large acreage in less time. It's cost effective. We can also have possible sprays during the season, and mostly when I show you the epidemiology, when we are going to have the inoculum present through a longer time on the growing season, so we may need to reapply that product. And of course, there is information required for registration of this product and different ways to apply. So just to give you an example, different countries and different regions will have already a significant amount of product registers. These are just in California, product register chemicals and biologicals. I'm sorry, I don't know exactly the current situation in Michigan and which products are registered, but different regions, have different products. So of course you have to confirm registration, in the region before use. I just wanted to give you an example of our current situation in Canada, where we currently don't have any product registered to control grapevine trunk diseases, nor chemical, neither biological. So because of the current high demand of biological options by the industry, right now, close to 20% of our acreage are farmed organically, and there is a program on sustainability in BC. The industry was really interested in developing these local biological agents. So one of my master's students, Jinx, have been working first in identifying biological control agents from British Columbia. We have identified 8 different species of trichoderma. And as you may know, trichoderma is one of the most commonly known biological control agents with antifungal activity. And we have performed both laboratory as well as greenhouse in order to select the best isolates, to perform in our field trials. So quickly, we have just recently completed our second year of field trials here in British Columbia in a Merlot vineyard, where we prune our vines to 3-4 nodes. So basically, the idea is, because we are artificially inoculated, when we come later and prune to 2 buds per, we get rid of the infection and it's not going to reach our cordon. So we have used, of course, our 3 different BC trichoderma-based products, where we use 50,000 spores of trichoderma per boon, and compare with commercial Yield Seal, is a tebuconazole product, or T77, which is a commercial trichoderma atrobiride. So we were comparing our products against these commercial products. We have been challenging these boons with 2 Botrytis pathogens, Diplodia seriata and Fusiculum parvum. And basically what we did is on April 10th, 2009, for example, we treat all the boons, and then we came 24 hours, 7 days, 21 days, and 60 days post-treatment to different mines to challenge with the pathogen, because we wanted to know not only the efficiency of our trichoderma and the commercial products, but also how long they will last in the vine. So this is just the results of our positive controls. So these pruning wounds were non-treated but inoculated with the pathogen. So you can see 24 hours after pruning, of course, the pruning wound is going to be highly susceptible. We obtain the highest infection. As long as the age of the pruning wound increase, the pruning wound itself is going to be healing and is going to be less susceptible. So we can see here that at 60 days, we have a significant reduction of susceptibility, but still infected. Natural infection, so those were cases from the vineyard. We wanted to know if there was any natural infection happening, and it was very, very minimal. So results from our trial in 2019, and here I'm representing at the mean percent disease control. So the longer the bar, the highest control. We can see here our yield shield commercial chemical product based on tebuconazole basically give us 100% control of these pruning wounds up to day 21, and decrease a little bit later on after 60 days. But still, after 60 days, we were able to get significant control. We have also to take into consideration that the pruning wounds after 60 days are less susceptible and have less risk of infection. Our biocontrol commercial T77, very, very high performance. And we can see that after 24 hours, they were able almost to reach over 80% and very consistent through the 60 days. Our 3 different treatments, very good results. We're very happy to see that most of our products, even after 60 days, were comparable to either the chemical or the biological commercial control. So they have both very high control, and we were able to keep our pruning wounds protected over 60 days at a higher 90% rate. So very, very good success there. So as I mentioned before, in other parts, depending on the work we do on epidemiology, we can manage the pruning in trying to minimize the impact of these grapevine trunk diseases. For example, in California, as I mentioned before, late pruning is one of the options they have. However, here in British Columbia, late pruning is not feasible because most of our inoculum will come, at the end of winter, early spring. If we do late pruning, we are basically opening the wounds closest to our first inoculum. So our question is, can we manage the pruning in British Columbia by, having an early pruning? So we have a study where we were pruning different vines. This is a Chardonnay inoculated with Neophysaceae parvum with 5,000 spores. So basically what we did here is certain number of vines were pruned in December, others in January, others in February, others in March, and others in April. And April 16, one day after the last pruning time, we artificially inoculate all these, oops, sorry, this is mine. We're able to inoculate all the pruning wounds, and we can see that, for example, in our case, by pruning early in January, we were able to reduce up to 80% infection compared to pruning, for example, March and April. The later we prune in BC, our inoculum is going to come right after, the pruning wound is done, which is going to be highly susceptible. So it's going to have a much higher risk of infection. So, with an early pruning, we are at least minimizing this infection. So as conclusion, management of grapevine trunk disease needs to occur, from nursery to vineyards. Importance of effective and robust diagnostic tools. We need to improve the health status of the planting material and really understand the role that biotic and abiotic factors may play on disease development. How to properly, plant and manage those first years of the vineyard in order to avoid that these young bite decline diseases develop in our blocks. Need to understand epidemiology, and this is very important, to understand the high and low risk periods of infection through the year. And we need, as we saw, because the pathogen is in the environment for a long time, protect our pruning wounds, either by chemicals. Don't underestimate biological products. We are having, really, really good results with some of the biological products, which have shown to protect wounds up to for 60 days. And pruning time can help to reduce the infection. In our case, in British Columbia, just an early pruning may reduce infection up to for 80%. Vineyard sanitation continue to be important, and of course, we need to start thinking on controlling plant diseases the same as we control powdery mildew. We need to start since year one. Since that first pruning we do in our block, we need to start thinking in avoiding the entrance of this pathogen. So basically, it's really a holistic approach where not only one control is going to solve everything, but following good IPM recommendations, having good chemistry or good biological control agents, knowing exactly when to apply them, when it's going to have the most efficiency, is going to have really a big difference of maintaining your vineyards a longer time. So I just wanted to show, big literature review that we published recently in plant disease, where a lot of my presentation today and some other things can be found. So it is quite a piece of information for you to follow many different control practices. And with that, I really thank our funding sources, as well as my lab here in Summerland in British Columbia. And with that, I'm very sorry for the technical issue. I was able to finish at least within the hour. I have time. I'm here for questions, so hopefully we can have the time. I would like to thank also Michigan State University and Ismail for the invitation. A pleasure to be here. And of course, a Zoom meeting without technical problems is not a meeting. So thank you very much.
Thank you, Dr. Torres. Thank you very much. And sorry for the technical issue. And thank you for joining us again. So
it was probably my end.
It was perfect presentation. Thank you so much.
Yes, thank you. We do have some questions for you, Dr. Urbez-Torres, if you have a moment.
Yeah, I have the question and answer.
Okay. Someone has asked, why would sudden vine collapse only cause the virus to sometimes and recover other times? I think what they mean is sometimes recover.
Yeah, so this is a very good question. And unfortunately, even though ESCA disease, so this phenomenon happened with ESCA-infected vines and what we call apoplexy, we know that some of these vines collapse in the middle of the summer or where the highest needs of water from the plants are. We still don't understand. As I mentioned before, ESCA is one of the longest studied disease. And there's a lot of work right now trying to understand the physiology, but we don't know why some of those vines really are able to recover. Some of them, they don't recover. Some of them, they collapse, all the leaves wilt, and the fruit, of course, drop, and they will not recover the following year. It could be depending on the amount of infection inside the plant, but there are also another physiological circumstances that is being well studied in France. How this hydraulic conduction within the plant and how the water is transported on these vessels where they are infected versus not infected. I'm sorry, I don't have an answer for you. It's still very, very complex disease. We don't understand, even though we have been studying for a long time.
A follow-up question to that is, if the vine does recover, next season, is it prone to suffer the same fate?
Yes, if the vine is recovering the same season, what we see is that vine probably shows a lower vigor, and it may eventually, collapse again that year. Sometimes those vines, they don't collapse the year, but that vine basically is weak enough that eventually, it will collapse. So it's not a total recovery, and the vine will be fine again.
Okay, thank you. A reminder to our attendees, if you have a question, please type it in the Q&A box at the bottom of your screen. Another question, you have discussed fungal pathogens. Do you have any comments on Agrobacterium yes, causing crown gall, which is a common issue here in Northwest Michigan?
Yeah, so we have the same issue in British Columbia. And it's the pathogen causing crown gall on grapes is Agrobacterium vitis. Tumefaciens cause crown gall on tree fruits. So Agrobacterium vitis is very, very specific to vines. We have the same problem here in BC. crown gall is not considered a grapevine trunk disease by definition because grapevine trunk disease are caused by fungal pathogens, but I understand that it's also causing, problems in the trunk. I personally don't have right now research going on crown gall. There is a group at the University of British Columbia here in the Calgary campus. They have a project on crown gall, and they're actually working on trying to both look for alternative in vineyard cultivation as well as biological control to minimize the problem with crown gall. And one of the problems we have with crown gall in our cold regions is, the bacteria is in the plants. It has been shown that the bacteria can come is naturally in the plants in many occasions. And when we have, a break due to winter conditions, the split of the vines, the crown gall is activated and the galls happen. And even if the plants are not coming with crown gall from the nursery, if they are planted in an area where they have been grapes, crown gall probably is still on the ground and it can infect again. It's a very difficult disease to control and to manage and to deal with, and we suffer the same problem here in BC.
Okay, thank you. I think maybe you addressed this in your presentation, but someone wants to know how soon after pruning must a spray be applied to provide effective control?
So as I say in my presentation, it's going to be, my advice is, as soon as possible after pruning, you should apply your fungicide. However, having knowledge of the epidemiology, those graphs I shown that when your inoculum is going to be present in the area, it has really, really valuable information. For example, we have here, of course, growers start pruning in January. We know that the months of December and January, the amount of inoculum in our vineyards is very, very minimum. So, and because of the cold conditions, and we know that the spores cannot germinate, you can prune those vineyards and maybe wait until later, to apply the fungicide because you have this window of time until the inoculum comes. However, here, those growers in British Columbia that they are pruning later, closer to March or April, at the end of the winter, early spring, they should apply the fungicide or the biological control immediately after, because at that time, we know that as soon as the temperature rises above freezing temperatures, 5-6 degrees Celsius, that's what the spores need to germinate, and we have some precipitation, we are going to have that first inoculum coming. So those wounds, they need to be protected. There are some products that also have a curative effect, meaning that if you prune your vineyard and you get infection, let's say 1 or 2 days after you have pruned, but you are able to put and spray the third or fourth day, that spray will kill the spores on the surface on the pruning wood that they start germinating. So it's not a very long timing. Studies in Australia have shown up to 7 days. You have basically to put that spray in your pruning wounds, so you are sure that if any infection has already started, that first spray on the wound is able to eliminate it. It's going to be depending a lot on the growing rate.
Okay, thank you. Another question. To extrapolate results to our site, would you say that pruning should be complete prior to meant temperature of 10 degrees Celsius, or what cutoff temp would you recommend?
So as I saw before, we know in our conditions, and we may not be probably as cold as in Michigan, we start reaching this above freezing temperatures, I would say after mid-March, we start reaching these 5-6 degrees. So if we are pruning earlier, as I mentioned before, where the temperatures are, low, we know the pathogen cannot germinate, the spores cannot infect. So 10 degrees is actually, 10 degrees is Celsius is a considerable temperature already where the pathogen can be very happy and can start germinating and infecting our pruning wounds. But also we have to take into consideration that most of the spore release happen in correlation with precipitation. So this is something also that growers, keep an eye when they are pruning, and depending the time, some growers are able to delay a few days to avoid this precipitation. Many growers cannot. So that's why at the end, the treating of the pruning moods is going to be critical.
Okay, thank you. And these are some great questions. When you speak of vineyard sanitation, are you talking of removing the prunings from the vineyard floor, and any other expansion on your ideas on vineyard sanitation?
Yeah, so vineyard sanitation, is a good question. I didn't have time to expand on it. Vineyard sanitation can go from eliminate infected parts of your vine. So for example, you have already, a cordon or spare positions that you are noticing that they're already infected. As I saw in the video, all infected regions are gonna start developing the fruiting bodies of the fungus, which is gonna have the spores inside, and it's gonna be releasing those spores into the vineyard. So removing these infected parts, is one of the first steps. There is significant research done that pruning debris left on the ground, it can actually harbor a lot of these fruiting bodies, and it can be also a source of infection. So removing that pruning debris out of the vineyard is recommended. How and is sometimes difficult, to, for example, in some regions, we have difficulty to get burning due to environmental conditions. So there is less and less options to remove that material and burn it. But for example, growers, what they do is they chop and they reincorporate sometimes into the rows and try to bury that prunings. So those are the most common sanitation. In California, it's very common through the dormant season and some growers as well here in BC do apply lime sulfur, as a dormant, basically disinfectants and by contact, will kill basically most of these fruiting bodies if they get rich on it. So that's part also of sanitation procedures that some growers do. So so these are I would say that the main sanitation practices.
Okay, thank you. What nutrients are most important to keep vines healthy regarding trunk disease?
This is a very good question. I don't think we have really a study of specific nutrients, that to keep healthy from trunk disease. I would consider trunk disease as any other disease. And when you are developing your vineyard during the first years, any practice you do, not only on nutrients but in irrigation, that minimize the stress of the vineyard. They should be applied. I don't have a specific in terms of which type. I don't think we have a specific nutrient, that will help the vine against trunk diseases, unless at least that we have a study or researchers. So the main goal is try to avoid this stress, whether it's biotic or abiotic stress. It's important to know that some, also excess of fertilization can be detrimental, for the vines. If we have a lot of growth, we can be promoting powdery mildew, for example, with a lot of exuberance and more growth. We can, in our regions, for example, we can delay leaf fall, which, some of the issues for that would be that the plants are not getting dormancy, on time ready, so we can have some winter kills. So I would say that for trunk diseases, we don't have any specific. You follow your guidance for keeping the vineyard, as healthy as possible and minimizing the stress.
Okay, thank you. And thank you for all of you for sticking around. I know we had a little technical difficulties and we're going over time, but
Sorry about that, yeah.
That's okay. These are some great questions. So we'll keep going for those that can hang on. And you too, Dr. Urbez-Torres. The next question is, does the healing over of early pruned vines aid in preventing infection of fungal pathogens? Do peroxide-based chemicals aid in prevention of infections?
So the pruning wound of the healing of the pruning wound is a natural process. That we know that basically this process get accelerated as with warmer temperatures. So the warmer it gets, the faster the pruning heals. That, for example, one of the studies conducted in California, late pruning. That's why we are also minimizing the infection, because those pruning wounds are healing much faster. However, if you prune probably in our conditions, Michigan or BC, in the middle of winter, December, January, that bone is going to take much longer to heal. It's going to be, they don't have that physiological response to start healing. However, because it's taking longer to heal, but it's also during a period that we know that the inoculum, is much lower, and there is not inoculum due to our environmental conditions. We feel that that's the main reasons why those wounds are not infected. And during that period of month and a half, of 2 months, this healing of the wounds start happening. Regarding the application, I don't have right now information regarding on trans diseases, with application, and if this will favor minimizing the infection. But I could hypothesize that any product that speed the pruning, the healing of the pruning wound, could be efficient of minimizing the infection. But unfortunately, I'm not aware of studies done on that in trans diseases, and I cannot have a clear answer for that type of products.
Okay, great. We have a question of, when would you recommend termination of pruning in Michigan?
I would like to probably, and I should have probably studied more the conditions, in Michigan, but based on the studies done on Yutai palata sport trapping in Michigan in the 80s, and as I showed, most of the captures of the sport happened March, April, May. Those are the 3 critical months that, correct me if I'm wrong, probably you are experiencing the same conditions than us. You are coming out of the winter, increasing temperatures above freezing. It may be some precipitation, so that's why probably your most of the inoculum is happening during that time. So I would say that it's gonna be difficult probably to go far beyond those months in Michigan, more than April, May. I'm not 100% sure where mostly bud break happens in Michigan. I don't know how long you can wait until pruning in Michigan. But I would say it's a very similar situation than BC. I think the pruning should be done, probably before that time. And same as in British Columbia, those pruning wounds, they will need to be treated, because you are not going to be able probably to delay as much as possible to go away of that period of sports. You will go entering in May, June. I don't know if it's still possible there. I don't think so. So it will be basically like here, either pruning earlier. I don't know if the possibility of pruning earlier in Michigan. Maybe the temperatures also are. Sometimes there is concern that very low temperatures could affect the buds once they are pruned. Some growers here, what they do when they prune earlier, they usually leave an extra bud, to make sure that hopefully, they are not going to have issues with winter damage or spring frost. So it's a tough one for me, because I'm not very familiar with the conditions in Michigan, but I think it's pretty similar to BC. I think there's probably a possibility to prune earlier and try to avoid, that period of infection. I don't know your snow cover on the ground. That's a big factor. Sometimes we cannot enter in the vineyards here because we have more than a foot of snow. So that's also a big factor for pruning, during the winter months. So I think you are in the same boat. Probably you're gonna have to prune, the usually February, March, probably April, and you are gonna have to make sure those pruning wounds are covered based on that study with Utaipa.
Doctor, sorry to interrupt. I'm going to share my screen, and because I'm going to share the code for grower here. I don't want, just want to make sure everybody. So you can go ahead and answer the question if you have time.
Another question is, in northern Michigan, if we prune in winter when inoculum is low, aren't the vines more susceptible to frost damage?
Yeah, so exactly that's what I comment.
Oh, I
And that's
apologize.
one, yeah, that's one of the concerns that growers have also here. If vines will be more susceptible to frost or even to winter damage. We haven't done specific studies on that, and probably some of the more viticulture people would know better the physiological process of the vine, and the response to that. But we have been pruning here in BC. Usually, the big growers with big uh, vineyard blocks, they usually start pruning in December. Those blocks, and we don't usually experience that problem. We can reach temperatures here minus 20 Celsius, minus 22 Celsius. It's true that those temperatures are for a short time in the night. Usually, if we have snow cover, but I mean, we see winter damage sometimes, but not as much issues with the early pruning and spring frost. But again, that's another area I think it needs more research in regards of trunk diseases based on what we are talking. We are talking right now how to manage the pruning and the different periods that we can prune. So now we need to look further and see, well, yeah, I know I can minimize the infection of trunk diseases, but also what that means in terms of my vineyard and production. I think those answers need to be aligned with that.
Great, thank you very much. We do have a couple of other questions that I think because we're running out of time.
I think we can send it via email.
Yeah, we'll send those via email.
Because we are, 12:33. So
for sure. And I just wanted you to know that there were many comments in the chat about thank you for the great information and thank you for the great presentation. So
my pleasure. Yeah. I hope that in a short future, I'm able to visit Michigan and
of course, yeah, we would love to have you here,
and see the great production there. I never been, but yeah, thank you so much for the invitation, and I appreciate it.
Yeah, thank you so much for your time, and thank you everybody for joining Northwest Michigan Orchard and Vineyard Show. Great session, and I would like to say thank you to our support there, financial support there. They did great job. They helped us to prepare this show for you. And yeah, and these are our amazing collaborators. Thank you so much. And we would love to have you tomorrow in great session in 10:15. And also in 11:30, we will have P45 annual meeting. Thank you so much. I appreciate your time.